Dear students, today we are here to continue our topic, that is insulin. In this session, we will study about the actions of the insulin, its administration, adverse reactions, and preparation of insulin. So let us start with the actions of insulin. Pharmacological actions. First, we will study its actions on liver. Insulin tells the liver to bank as much glycogen as possible for later use. It causes increase in the activity of glucokinase. Glucokinase is an enzyme which causes phosphorylation of glucose. It increases the glucose uptake. Insulin increases the glucose uptake by cells from blood vessels. That is, it causes trapping of glucose within the cells. Next, it increases the glycogen synthetase activity. It increases glycogen synthetase activity. It increases glycogen synthetase activity, which causes increase in the glycogen deposition. Next, in liver, this was an increase in activity. Now, in liver, it causes decrease in the activity of phosphorylase enzymes. That is, which leads to the glycogenolysis. That is, it causes decrease in the activity of phosphorylase enzymes, which causes breakdown of glycogen. Next, it causes decrease in gluconeogenesis. That is, synthesis of glucose from non-carbohydrate source. This was the action on liver. Now, its actions on adipose tissue. In adipose tissue, insulin causes fat cells to become more permeable to glucose. That is, it increases the permeability. Increases uptake and utilization of glucose. That is, insulin causes increase in the permeability, uptake and utilization of glucose by fat cells. It leads to the synthesis of number first glycerol, alpha glycerophosphate, and fatty acids. This alpha glycerophosphate and fatty acids combine to form triglycerides. Triglyceride is a storage form of fat. That is, insulin causes fat synthesis, which we call as lipogenesis. Now its actions on skeletal muscles. In skeletal muscles, insulin stimulates the entry of amino acids into the cells, which promotes protein synthesis. That means in skeletal cells, it also promotes protein synthesis. This was about the actions of insulin. Now coming to the administration. Insulin is destroyed by gastrointestinal tract, so it should be given by subcutaneous injections. Now coming to the adverse reactions. Adverse reactions of insulin. Insulin causes hypoglycemia. Hypoglycemia is the decrease in the blood sugar level, which leads to the coma and convulsions. Hypoglycemia leads to coma and convulsions. Next effect is that it causes allergy. Allergy is mainly characterized by redness and swelling at the site of injection. It can also lead to the obesity, which may lead to increase in the body weight. Next, now we will study about its preparations. Preparations of insulin. Depending on the source, we have three types of insulin. That is bovine insulin, porcine, and human insulin. Bovine insulin we get from cattle, porcine insulin we get from pork, and human insulin is the insulin which we obtain by recombinant DNA technology. It has been observed that human insulin differs from the cattle insulin by 3 amino acids. Human insulin differs from cattle insulin by 3 amino acids and from porcine by 1 amino acids. This was the source of the insulin. Now we will study about preparations of insulin. In general, we have three types of insulins, that is short-acting, intermediate-acting, and long-acting. Short-acting insulin include plain insulin, and insulin zinc suspension. We also call it as semi-lengthy insulin. Another is this was short-acting insulin. In this short-acting, we have plain insulin, which shows its action for 8 hours. Next is the intermediate insulin. Intermediate insulin is globin insulin. Isophane insulin. And insulin zinc suspension. Zinc suspension, which we called as lenti insulin. Its action remains for 24 hours. These are 24 hours acting. So intermediate insulin include globin insulin, isophene insulin, and insulin zinc suspension, which we call as lanti-insulin, and its action remains for 24 hours. Isophene insulin is also known as NPH insulin. Next we have long-acting insulin. Long-acting insulin. Long-acting insulin includes protamine insulin. Protamine zinc insulin. Protamine zinc insulin and insulin zinc suspension. Insulin zinc suspension, which we call as ultra-lenti-insulin. Ultra-lenti-insulin, and its action remains for 36 hours. This was about the insulin. For further details, we will continue in next session. Till then, thank you.